Hello everyone, welcome to the fourth module of the paper Cell Biology. In this module, we will study about the ultrastructure of chloroplasts, which includes the chloroplast genome, sorting of chloroplast proteins and function of chloroplast, which includes the process of photosynthesis and cyclic electron flow. This module would also give us a fairly good idea about the similarities and distinguishing features between mitochondria and chloroplast. Structure and function of chloroplast. Chloroplasts, the organelles responsible for photosynthesis, are in many aspects very similar to the mitochondria. Both chloroplasts and mitochondria function to generate metabolic energy evolved by endosymbiosis, contain their own genetic system, replicate by division. However, chloroplasts are larger and more complex than mitochondria, and they perform several critical tasks in addition to the generation of ATP. Chloroplasts are only one of several types of related organelles that play a variety of roles in plant cells. Plant chloroplasts are large organelles like mitochondria are bonded by a double membrane called the chloroplast envelope. In addition to the inner and outer membranes of the envelope, chloroplasts have a third internal membrane system called the thylakoid membrane. The thylakoid membrane forms a network of flattened discs called thylakoids, which are frequently arranged in stacks called grana. Because of this three membrane structure, the internal organization of chloroplasts is more complex than that of mitochondria. In particular, their three membranes divide chloroplasts into three distinct internal compartments. The intermembrane space between the two membranes of the chloroplast envelope, the stroma which lies inside the envelope but outside the thylakoid membrane, and the thylakoid lumen. Despite their great complexity, the membranes of chloroplasts have clear functional similarities with those of mitochondria as expected, given the role of both organelles in the chemiosmotic generation of ATP. The outer membrane of the chloroplast envelope, like that of mitochondria, contains porins and is therefore freely permeable to small molecules. In contrast, the inner membrane is impermeable to ions and metabolites, which are therefore able to enter chloroplasts only via specific membrane transporters. These properties of the inner and outer membranes of the chloroplast envelope are similar to the inner and outer membranes of mitochondria. In both cases, the inner membrane restricts the passage of molecules between the cytosol and the interior of the organelle. The chloroplast stroma is also equivalent in function to the mitochondrial matrix. It contains the chloroplast genetic system and a variety of metabolic enzymes, including those responsible for the critical conversion of carbon dioxide to carbohydrates during photosynthesis. The major difference between chloroplasts and mitochondria in terms of both structure and function is the thylakoid membrane. This membrane is of central importance in chloroplasts, where it fills the role of the inner mitochondrial membrane in electron transport and the chemiosmotic generation of ATP. The inner membrane of the chloroplast envelope does not function in photosynthesis. Instead, the chloroplast electron transport system is located in the thylakoid membrane, and protons are pumped across this membrane from the stroma to the thylakoid lumen. The resulting electrochemical gradient then drives ATP synthesis as protons cross back into the stroma. In terms of its role in generation of metabolic energy, the thylakoid membrane of chloroplasts is thus equivalent to the inner membrane of mitochondria. The chloroplast genome. Like mitochondria, chloroplasts contain their own genetic system, reflecting their evolutionary origins from photosynthetic bacteria. The genomes of chloroplasts are similar to those of mitochondria in that they consist of circular DNA molecules present in multiple copies per organelle. However, chloroplast genomes are larger and more complex than those of mitochondria, ranging from 120 to 160 kilobase and containing approximately 120 genes. The chloroplast genomes of several plants have been completely sequenced, leading to the identification of many of the genes contained in the organelle DNAs. These chloroplast genes encode both RNAs and proteins involved in gene expression, as well as a variety of proteins that function in photosynthesis. Both the ribosomal and transfer RNAs used for translation of chloroplast mRNAs are encoded by the organelle genome. These include 4 rRNAs, which are 23S, 16S, 5S and 4.5S, and 30 tRNA species. In contrast to the smaller number of tRNAs encoded by the mitochondrial genome, the chloroplast tRNAs are sufficient to translate all the mRNA codons according to the universal genetic code. In addition to these RNA components of the translation system, the chloroplast genome encodes about 20 ribosomal proteins, which represent approximately a third of the proteins of chloroplast ribosomes. Some subunits of RNA polymerase are also encoded by chloroplasts, although additional RNA polymerase subunits and other factors needed for chloroplast gene expression are encoded in the nucleus. The chloroplast genome also encodes approximately 30 proteins that are involved in photosynthesis, including components of photosystem 1 and 2 of the cytochrome BF complex and of ATP synthase. In addition, one of the subunits of ribulose bisphosphate carboxylase or Rubisco is encoded by the chloroplast DNA. Rubisco is a critical enzyme that catalyzes the addition of carbon dioxide to ribulose 1,5-bisphosphate during the Calvin cycle. Not only it is the major protein component of the chloroplast stroma, but it is also thought to be the single most abundant protein on earth. So, it is noteworthy that one of its subunits is encoded by the chloroplast genome.